Hello everyone. Good evening, everybody. So today I'm going to talk about organoids and how these organoids are transforming our drug discovery, the paradigm of drug discovery, and how these organoids can be used in disease modeling. So these are the slide outline which I'm going to discuss. So first I will discuss about how one drug get FDA approval and get into the market, and how much efforts and cost it bears to bring one drug in the market. Then how organoid can help to expedite the drug discovery process, and how we can create organoids in our lab. Then advantages of organoids over traditional methods like 2D culture or something, or cell lines or animal models. Then application in drug discovery, disease modeling, precision medicine, organoids. Then what are the challenges and future prospects of organoids? Then session will be open for question answer round. So this is the general paradigm of drug discovery process and how one drug comes in the market. So it's a very costly process. Approximately, for one drug to come into the market, it takes billion and million of money. And if one drug fails in any of phases, like from it starts from target validation, compound screening, lead optimization, preclinical test, phase 1 clinical trial, phase 2 clinical trial, phase 3, and then final approval of the drug. And it's a very, and you can see how much time it takes. So approximately to come one drug in the market, it takes 10 to 12 years of hard work and money. So it's a very expensive process. And any drug, you can see in compound screening phase, the process starts with 10,000 candidates. That is, 10,000 candidates have been selected for to treat any disease. And if you can see in the last phase, that is the approval of drug. So you can see the funnel size, how it's reducing, and how drugs are failing. So, and how organoids can help in this drug discovery paradigm. So in the preclinical phase, there is it can be divided in two parts. First is a conventional method that is using cell lines, 2D cell lines. It enables gene and drug association study and various kind of drug toxicity. It's a mechanism of action. What is the pathways involved? All these study can be performed on 2D cell lines. And after 2D cell line, if some drug has passed toxicity analysis or genomic study, then it will go towards the PDO, or simultaneously 2D cell line study and three-dimensional patient-derived organoid study can be done simultaneously also. So using PDO, patient-derived organoids, or 3D organoids, genomic study, RNA study like transcriptomics, epigenomic study, and protein, drug sensitivity, all these study can be aligned with the drug discovery process. And once you get all these data, then you can identify patients. So which kind of patients you have to test. You randomly will not go and check the drug on any volunteer. So you can, according to, after getting the data from present derived organoids or the 3D organoid, you can do the clinical trial in a stratified manner. That is, you will not randomly do your clinical trial on anyone or anybody. You can make a group of patients, and then you can cut down the cost of clinical trial, because in drug discovery process, clinical trial is a very crucial and very costly phase. So it takes a lot of money and lot of efforts, and lot of people are involved in the clinical trial phase. So if you can somehow using organoid data, if you can streamline these processes and you can make the groups of specific patients, then it will be a more fruitful method. And finally, you can offer personalized medicine also. If some person is suffering from cancer and they are ER, PR, HER2, these are the breast cancer markers. And if they are, you can offer personalized medicine using the PDO method. So that will be the ultimate goal of organoid-based drug discovery process. Secondly, you can see in this slide that how much money is involved in this. So you can see on the left side of the screen, $1 million for 1 million compounds it starts with. And as you go progress in this drug discovery paradigm, so the cost is increased to $1,000 million, and you will get one compound out of that. So if in the primary screening, that is the first phase of the drug screening process, target identification. So in that phase, if you can couple your 2D cell-based assays with 3D based testings like toxicity, dose response, cross-validation, pharmacokinetics, and metabolism, all these lead optimization, if you can do with the 3D model. So this 3D model is more closer to human clinical trial, because it's directly coming from the patient or from the healthy volunteers. Sometimes what happened during drug discovery process, all the drug passes all these criteria. They show beautiful results in animal and mouse study. In monkey study, some of the vaccines get passed. And when this drug, after spending so much of money and time, when it goes for clinical trial, it fails because of the difference in species. There is a species difference between human and animal. So organoids are more closer to this human clinical trial. So if you can bridge this gap between in vitro and in vivo, that is animal study, with organoid, then it will be more fruitful. And in the drug discovery process, as I already mentioned, it's a very costly process. So if you fail fast, then you can save money, and the failure will be more safe, because you can save money, you can save time, and that same time you can utilize to get some more targets or some more drugs which could be more effective for the disease. So fail fast, fail safe. So this is how organoid can help. So next, how organoid can be implemented in drug discovery or drug screening process. So first, if we have identified some compounds using in silico studies, then directly we can take it for in vitro studies, that is on 2D cell line. And we can skip mouse, small animal testing models, and directly we can go for organoid study. And from organoid study, whatever we will get data, based on that data, we can go for clinical studies. Or in the same study, you can add small animal testing also, and you can get more robust data. And then you can implement that more robust data for clinical trials. So these two approaches can be implemented in drug discovery process using organoids. So now the question is how you will make these organoids, what kind of cells can be used for organoid generation. All these are the questions which I'm going to address today. So there can be three methods. First is human embryonic tissue, that is the blastocyst phase of the embryo. From there, you can isolate embryonic stem cells, and from embryonic stem cells, you can make spheroids. Or, because I know that embryonic stem cells have some ethical issues, because there is involved a fetal abortion. So in many countries, including USA, research on embryonic stem cells is banned. So what is the alternative of embryonic stem cells? So it's induced pluripotent stem cells. So this iPSC technology was discovered in 2012 by Shinya Yamanaka, a scientist from Japan. So what he has done during 2012, he has reversed the clock. And what he has done from directly, he has used some genes, and directly the human adult cells can be reprogrammed to iPSC. And from now this iPSC induced pluripotent stem cells. Pluripotent stem cells means it will act as a embryonic stem cell. So it means it can, the iPSC induced pluripotent stem cells can differentiate towards any lineage of the cells. It can make brain, it can make heart, it can make liver. So using iPSC, you can make all these kind of organoids. It's a Nobel Prize winning technology, and it's very helpful. This iPSC is going to change the paradigm of drug discovery process. Other method that also can be used, that directly you take patient's primary tissue and isolate the cells and make your organoid disease model. From blood also you can make some cancer cell lines or immune cells you can isolate, or even PBMC, peripheral blood mononuclear cells, cancer tissue. From cancer tissue you can make cancer organoid. So all these are the methods to use cells and develop and generate organoids. So there can be two types of 3D models. One is organoid and second is spheroid. But organoid and spheroid both are very different. In organoid, we can co-culture, mix many type of cells and make the small version of organ. It will mimic the organ more closely. Whereas in spheroid, there is only one type of cells. Like if I take the example of liver. So in liver, there are many type of cells like hepatocytes. Hepatocytes is major cells in liver. Then stellate cells, some macrophages, immune cells, blood cells, even endothelial cells. So in spheroid, you will take only hepatocytes, that is the 70% of liver, and it will mimic 70%, it will be closer to the liver model. But if you are going to culture these liver cells as an organoid, then you will need all kind of cells present in the liver, like hepatocytes, stellate cells, immune cells, macrophages, even endothelial cells to make the vascularization. So all these things you need for organoids. And organoid is more closer to the mimicking human body as compared to spheroid. But spheroid is easy to generate, or in other case, organoid is difficult to maintain and difficult to create. Everybody cannot create organoids. Spheroid is simple to generate and handle. Other thing, how we can create these organoids. So as I already mentioned, that there can be different types of cells directly from primary from patient tissue or healthy volunteer tissue. You can take the tissue and you can create the organoid, or you can take the fibroblast or PBMC and reprogram it to iPSC, induced pluripotent stem cells. And then you can make whatever organ you want. But from tissue, whatever tissue you will take, if you are taking liver tissue, then you will able to make only liver organoid. Other, how you will generate these organoids, so there can be different models: scaffold independent or scaffold dependent. Scaffold independent means you are not giving any supporting matrix to the cells. So it can be using a spinning bioreactor or hanging drop, where you will put this put your cells, mixture of cells as a hanging drop, as you can see in the picture. And otherwise you can use low attachment plates, and you can seed your cells. And iPSC has this tendency to make a spheroid or organoid on its own if you will culture those cells in low adherent plate. And in the scaffold dependent method, you will mix the cells in with the hydrogel, matrigel, or any kind of extracellular matrix, and you will support these cells with the scaffold, that is extracellular matrix. And what kind of organoids you can generate? So till date, in the scientific world, we have generated brain organoid, pancreatic organoid, kidney organoid, retinal organoid. In retinal organoid, you can see there is a black retina, which is closely mimicking the retinal structure. Then in intestinal organoid, gastric organoid, these organoids are small miniature of all organs. So you can create all these organoids and you can develop human on a chip. So what are the applications? So there are multiple applications of these organoids. It has application in developmental biology, host microbe interactions, disease modeling, drug evaluation, as I already mentioned in drug discovery paradigm. Then transplantation, it can be used for regenerative medicine also, personalized medicine for cancer. Cancer is a very heterogeneous disease. And then cell therapy. So how you will make, what are the components needed for this organoid? So first, cell resource, as I already mentioned, there can be two type of cell resource. One is tissue. From tissue, you can develop organoids. And for this, you need patient or healthy volunteers biopsy sample from human, or it can be generated from animals also. For iPSC, you need any adult cell and then reprogram it to induce pluripotent stem cells, and you can generate organoids from iPSC. Then second, you have to give a physical cues, that is physical microenvironment, to these isolated cells. So it will be extracellular matrix support and signaling cues. So it will need some growth factors, signaling molecules, so that it can mimic the nutrient and waste diffusal of the basement member that is present in vivo. In the body, whatever ECM or cellular support is present, you have to give this in the in vitro, these in vitro models. And dynamic, if in our body nothing is static, but if you will see these 2D plates, it's a static culture where the cells are just sitting on the plate. But in if you are going for organoid or something, then you can provide dynamic tunability microenvironment also. You can culture it as a perfusion culture. It will mimic the blood flow in the body. Then growth factors. There are many types of growth factors, which is according to the organ's specific requirement. You have to add these growth factors to the organoids. So like for adult cells, you will need Wnt, EGF, epithelial growth factor, hepatocyte growth factor, IGF, FGF. For iPSC, for differentiation, you need activin A, BMP4, Wnt, FGF, VEGF, BMP, TGF, all these kind of soluble factors based on the organoid which you want to generate. Then matrix, there are multiple kind of matrix available in market, but you have to choose this matrix very carefully. It will also depend on your choice of generating the organoid. If you are going to develop a liver organoid, then you will take the matrigel. Matrigel is a liver ECM, or collagen. Collagen is a general ECM. Then there are many synthetic hydrogels are available in the market. So this matrix also you have to choose according to the organoid you need to generate. Then there is a integrating cues. As I said that in 3D organoid culture, you can generate blood perfusion, that is flowing of the blood you can mimic. So for that, you can use organ on chip models or multi organ on chip models. So here you have to integrate physiological and structural features of the organ. And by bioprinting, you can create your organoid of interest and you can produce the tissue for drug testing process. So these are the different organoids that have been developed, and for different disease models, these kind of organoids are used in drug discovery process. So there are many types of cerebral organoid, that is brain organoid, and it can mimic different types of disease like autism, schizophrenia, Zika virus infection, Alzheimer's disease, Parkinson's disease. Then optic cup organoids have been developed to study glaucoma or retinitis pigmentosa disease. For liver, organoids have been developed to study hepatitis, cystic fibrosis, coronavirus, because coronavirus is also infecting liver. So coronavirus diseases. For pancreatic, there are multiple applications like diabetes, cystic fibrosis. For inner ear organoid, some mutational disease of TMPRSS3 mutation study have been performed. For skin diseases, there is a skin organoid which can be used for drug discovery for the diseases like atopic eczema, then inflammatory skin diseases, sclerosis, systemic sclerosis disease. Then scientists have developed heart organoid also to study myocardial dysfunction. You can see the heart attack on the plate. So heart attack model has been developed using this iPSC lines. Then lung organoids have been developed and used to study respiratory disease. Then there is kidney organoid, which is used to study nephronic syndromes or polycystic kidney diseases. Then there is gastric organoid, which is used to study gastric infection in gastric organs. Then intestinal and colon organoids have been developed to study cystic fibrosis or any kind of microbial interaction. Even it has been used for coronavirus disease study also. So this is the one short snapshot of how these organoids can be used in drug discovery process or in disease modeling or drugs biobanking. Even these tissues can be biobank also. You can from different kind of patients, you can collect these tissue and you can make your biobank. And whenever you require to test any drug for any specific disease, you can take out this sample from organoid biobank and you can use for drug screening. Then developmental biology. Sometimes this disease occurred during the fetal development when the baby is inside the womb and some genetic mutation or some kind of injury that causes some developmental problem or some developmental problem in the brain or heart. So that also you can study using this iPSC technology, because when you are differentiating iPSC towards any organoid, brain organoid, liver organoid, so it mimics the developmental stages. So you can use this iPSC based organoids for developmental study. Then there is precision medicine. Precision medicine, as we know, can be used for cancer because cancer is a very heterogeneous disease. Every cancer patient is different from other patient because their cancer carries some different kind of mutation. Even in one person, if someone has breast cancer, so that cancer also varies position to position. If there is metastasis, so in the same patient, breast cancer will be different and the mutation can be different. And in stomach cancer, it will be different. It will express some other kind of mutation or some other kind of genes, which so its mechanism of action will be different. So in that case, you have to take samples from both sides of the cancer and you can create organoid, and that can be used as a precision medicine. Even if these data is utilized properly, then cancer patients which are having same symptoms and mutation of the organoid that have been developed in past can be that data can be used for the future patients. So it's a very great technology, precision medicine. Then drug screening, as I already mentioned, that these organoids can be used for high throughput, means many multiple timelines, multiple test points you can study in one plate, and all the organoids will behave similarly. So it's a very good tool for high throughput drug screening. Then regenerative medicine, where you can develop these organoids from the patient or healthy volunteer, and this can be used to repair tissues, like if there is some brain injury, heart injury, even for bone injury. Because once someone's brain had got injured or heart has got injured, their regenerative capacity is very low. So if from outside we can give them brain cells or cardiac cells, then it can repair their organ faster and better. And disease modeling by introducing CRISPR-based editing or by embedding immune cells in these organoids, you can mimic the disease more better on the plate. So these are all the applications of organoids, which I'm going to discuss in my later slides. So this is the complexity, model complexity. So cell line is very low complexity models. Cell line, because it's contain only one type of cells. If you are studying cell lines for liver cells, then HepG2, only HepG2 cell line will be there. Then from cell line primary cells, that is if you are isolating cells directly from the liver, so these hepatocytes will be more complex and more close to the human organ. Then 2D cultures, that is, you can set different types of perfusion or any kind of 2D culture plates. Then there is organoid, which is more closer to human. But the many things like in vivo conditions, you cannot mimic with organoids. Then there is animal models where you can study many types of mechanisms using these animal models. But only problem with animal model is there is a specific difference which with human. And then human, the ultimate goal for all these disease drug discovery process. Human is a more complex model to study, which we use in the final stage of drug discovery process, that is clinical trials. So what are the advantages of all these models like 2D cell culture and 3D organoids and animal models? So as I already mentioned, in 2D cell culture, there is no vascularization, means blood vessels are not there. So it's very uncomplicated and very initial phase if you are going with 10,000 drug candidates. So first, if you want to check just drug toxicity or any simple experiment you want to plan, then you can use these 2D cell lines. And it's very good for high throughput drug screening. Means you just culture these cells in a 96 well plate or 245 well plate, and you can get multiple checkpoints, multiple time points, many data you can get with this simple 2D cell culture model. But there will be no organogenesis, means it will be not show, it will not going to show any tissue function or nothing. It will just show one line, one cell line, its morphological changes, its toxicity study, all those things. Here you cannot mimic patient's original model for tissue engine for drug discovery process. And manipulation, yeah, you can do manipulation with these cells. You can introduce any mutation, or you can deprive these cells. You can play with the microenvironment of these cells. In modeling for human physiology, it's very limited because it's only one cell line. Then heterogeneity is very low because all cells are same. We are not co-culturing it with other cells. So, and it will be more homogeneous result it will give. And reproducibility will be high because it's a one type of cells, and it's a homogeneous population. So you can reproduce these results. And modeling cellular and mechanical communication will be feasible with these cells. Whereas in 3D organoid culture, you can create vascularization using endothelial cells. So you can introduce endothelial cells in this organoid, and you can create more closer models to human. And but it will be limited because you are not generating because the normal in vivo conditions are not you will not able to create in these organoids. You can biobank these samples, you can make a bank of these organoids. It's also feasible to high throughput screening. You can generate organogenesis also because you can generate isogenic tissues and transplantation. You can directly take the samples from the patients. So it will more closely will mimic the patient-derived organoid model. Then you can gene edit these organoids, manipulation or something. Then modeling, it will more closely will mimic the human physiology. Then heterogeneity is very high because different types of cells are there. Reproducibility will be low because you cannot produce same type of organoid always. And modeling and mechanical communication is feasible, whereas in animal model, as I said, there is multiple advantages, like it's feasible, it's mimicking the immune system, all those things, but high throughput drug screening you cannot do, and it's a very costly process, and there is a specific different species difference also. And patient-derived cancer organoids. As I already mentioned, that cancer is a very heterogeneous disease. So you can develop patient-derived organoids means if a patient goes to the doctor, so what doctor has to do, they just have to take the biopsy, patient biopsy, and that using that biopsy, organoid can be created, and multiple studies, drug screening trials to you can store these organoids also, and multiple study can be done on these cancer organoids, and these cancer organoids after taking from the patient will give multiple data, like for genetic editing. Then xenograft is a in which in more animal you will inject the you will transplant the patient tissue, and now it will become your cancer model, and in that animal you can test the drugs, and whichever suitable drug you find from the PDX model, you can refer that drug to the patient. Then drug screening, genome sequencing, you will get a lot of genome study data. Then it can be used for immunotherapy like CAR T therapy, and all these evaluate these therapies also. So if I am going to compare cell lines, PDX, as I already mentioned, PDX model and PDO, then it has their own advantages and disadvantages. Then there is advantage and disadvantage of these models. So I am going to focus on clinical trials. So clinical trial is very efficacious if it is used on the directly on human. But there is a chances of drug resistance also. Then pharmacokinetic study you can get from clinical trial. Adverse reaction, if some drug is showing some adverse reaction, though. So human is the clinical trial is the main best model for that. Then route of administration, where you have to give the drug, whether it's intravenous, intravascular, whatever. So for that, clinical trial is the best model. Then it's highly clinically relevant model, but at the same time it's very costly process. Then organoids in regenerative medicine, as I already mentioned, that from the from the patient's iPSC or some iPSC, or from the same patient's iPSC, you can store those iPSC, and whenever required, you can make the liver model or heart model, and you can inject. But it's a very tedious process, and it's under research, and you can replace heart, brain, or any other organ of the patient. Then generating iPSC, as I already mentioned, that you can make many type of organs using iPSC cells, and using these growth factors as listed in this slide. Then I will discuss about brain organoid. As you see, there are multiple brain diseases like Alzheimer, Parkinson's, all these disease, but very few drugs are available, because in if you will go for drug discovery process, then there is no models to test these drugs, which is the main limitation of using testing these brain drugs. So what people are using these days, that they are creating brain organoids using iPSC line, and those brain organoid mimic, which is more closer to the human, can be used for a drug discovery process. Then there can be two approaches for disease modeling. First, you can take healthy volunteers' skin biopsy and reprogram it to iPSC, and by CRISPR gene editing tool, you create some genetic mutation or something, and you can create your disease line. And in other side, second approach will be the patient who is carrying some mutation, you directly take biopsy from that patient, and you can create your organoid, and that organoid you can use for PDX, xenotransplantation, neurodevelopmental studies, genetic studies, vascularization also you can create. So these are two different approaches for disease modeling. Then infectious disease modeling. If you are studying any drug, which is like, if I'm taking the example of COVID. So when some person is getting COVID, then their body starts functioning differently, and it will create some inflammatory cells, some inflammatory secretion will be there. So that you cannot study on the one type of cell line. So for that, you have to create these organoid model, or if for vaccine testing, for example. So for vaccine development or any disease drug efficacy study, you need a organoid where you can embed your immune cells, that how these immune cells are going to fight against COVID. ECM, suitable ECM will be provided, and then you can introduce this organoid with pathogens also, like